Okay, let's please turn off our cell phones as we speak. Okay, now we will look inside the cell. Okay, we have a skeleton the same way the cells also have a skeleton. So, but what do we call a cell? Cyte means a cell. We looked at cytology, right? The same way, when you say cytoskeleton, it's the skeleton of the cell. But this skeleton, the framework is made up of various filaments. You have different kinds. They call them microfilaments, intermediate filaments, and microtubules. There are three kinds of filaments that help to make up the cytoskeleton. And the composition may vary, the kind of protein that makes each one of this. Like if you see microfilaments, they are made up of one kind of protein called actin. And if you see the microtubules, it's made up of tubulin. Tubules, tubulin. So sometimes some words help to connect the two different terms. So they play a role in giving, holding the cell in a certain shape and also guiding some of the organelles and other cell products to move from one compartment to the other compartment. Okay, so that leads us to organelles. Okay, so what are organelles by definition?
Tiny organs inside that perform a specific function
Right.
inside.
Membrane-bound compartments within a cell that perform specific functions. But in your book, it may be worded differently. An internal structure of the cell that carries out specialized metabolic tasks. But they're all surrounded by one or two layers of the same kind of phospholipid membrane, like your cell membrane or plasma membrane. So that's what gives it a unique advantage in reference to carrying things from one organelle to the other, because the membranes are of the same phospholipids, so they can merge with each other. So that should help you to recall the endo and exocytosis. Okay, remember the vesicles moving from one to the other compartment. So they merge and release the content one way or the other way, depending on which way it does. You call it exo and endocytosis. So the primary organelle is your central structure in a cell, which is your nucleus. The nucleus has certain parts, like your cell membrane. You have nuclear membrane, and there are tiny holes that allow things to move in and out. We call them nuclear pores. It's all in your book. You have looked up. And then you have the genetic material, which is your DNA. It's condensed and arranged in the form of chromosome inside of the nucleus. And you'll find another structure called nucleolus, which we need in order to synthesize the ribosomes that we will need later on to make your proteins. So all these are inside in a fluid. The fluid in the cell, what did we call that? Cytosol or cytoplasm with the organelles. So here they call it like cytoplasm, they call it nucleoplasm, all the content inside. So the chromosome, when you think of DNA, you have to be able to relate some of these words, because otherwise you may get confused. DNA, chromosome, chromatin, chromatin. These are all one and the same thing, but structural differences. The DNA is the basic material, which is the double-stranded structure which can go very long if you stretch it out, but when you roll them up, you call it chromosome, and it gets more condensed and thicker when you are ready to divide the cell during mitosis. That's when they call it chromatid. And always chromosomes occur in pairs, so they look alike, identical chromatids, one from the maternal and paternal source, so they also call them sometimes sister chromatids because they are identical. And there is another term, chromatin. Chromatin is nothing but when your DNA is arranged into a condensed structure, you need something to wind them around, so there are some proteins. So the proteins plus the chromosome together, they will call chromatin. I think there is a picture somewhere later in the next chapter that shows all the structural organizations of the DNA, how the DNA itself is arranged and how it's built into the chromosome. I wanted to mention that because you need to know the differences. And where are your genes? Genes are the DNA sequences that correspond to a protein. Those are the genes, if you have heard of genes. Okay, so for the nucleus, you should know the different structures that make the nucleus, and also the main function. It holds the genetic material, and also it controls all the cellular activities, because the genetic material serves as the blueprint for the cell for all the cellular functions, including synthesis of proteins and all the components needed for the cell structure and function. Red blood cells do not have nucleus, they are anuclear, but most of the other cells are nucleated, meaning they have nucleus. So you can see that in the very first paragraph. Then the next section discusses the endoplasmic reticulum. So we have membrane-bound channels that are necessary for protein synthesis. We abbreviate them as ER. These are your ribosomes, the tiny structures. So when the ribosomes are associated with the ER, meaning protein synthesis happening right then and there. So when proteins are being made, you need the ER and the association of ribosomes. So when the ribosomes are attached to the ER, you call it rough ER. And if you don't have the ribosomes attached, then there's your smooth ER. Smooth ER helps with some lipid synthesis and also is primarily engaged in detoxification. So if you see the discussion under smooth ER, you'll talk about detoxification of unwanted chemicals in your cell. So you can see the figures in 328 for the ER, which side has those dots that represents the rough ER. And you can see the microscopic images of the rough and smooth ER. So if you see the transmission electron micrograph on the top left, you can see the tiny dots on the outside of these channels. It looks a little rough compared to the one on the right. That's why they call it rough, and the other one the smooth ER. Can you all see it's very fine, okay? So the drawing is based on what is actually seen under the microscope. The TEM image, you'll see these lines. Those are your channels of the ER, and then you see the dots on the outside, if you look carefully. Ribosomes are basically made up of some RNA and some protein, and they are found in the cytosol, and they are needed to make the synthesis of protein, and they will come and attach to the ER. But one thing we need to know fundamentally you have the DNA that has to make one kind of RNA called mRNA, messenger RNA. You will see that in detail in the next chapter. And then that is used as a code to make your proteins. So once the dRNA double strand is copied and you make the messenger RNA that comes out through the nuclear port, sneaks out, it's attached, the ribosomes attached to them, and they bring that to the ER, and then your protein is synthesized and released into the ER. Then they come out in the vesicle, and then they go to the Golgi, the next set of channels, and then it goes to wherever it is destined to go, or wherever it is supposed to be.
So mRNA is a code to make the protein?
You need mRNA. Your DNA is just the blueprint, but based on that you have to make another molecule. It's one of the several types of RNA. There is tRNA, rRNA, mRNA. So mRNA is like a copy of your blueprint, meaning based on the DNA template, you make another set of sequences which are similar but not quite exactly. The composition of the mRNA is different in terms of the nucleotides, and then based on that, every 3 nucleotides in the mRNA will be representing one amino acid in your protein. So basically everything is in your DNA, but it goes through one more step. They call this transcription, and all of this is in chapter 4, so we are not there yet, but I just wanted to introduce. The next step is translation. Then the Golgi complex is similar to the ER. It's a set of membrane-bound channels, and then they help to process your proteins. The main function is packaging, modification, and delivery of your proteins. Those are the primary functions of your Golgi apparatus or complex. There is some processing done in the ER before they get into the vesicles, but the vesicles come and merge with the Golgi apparatus, and then it's further processed. Some proteins are just left alone. Some they add some carbohydrates, they become glycoproteins, and so on. And then it's again packaged into the Golgi vesicles and then sent to the destination. Besides that, Golgi also helps with carbohydrate synthesis. Whatever the cell needs, the sugar needs of the cell is met by the Golgi. Then to clean up the cell, you have lysosomes and peroxisomes. They contain enzymes that degrade unwanted material. So if you have a solid particle that is not required for the cell, the lysosome will come around it and phagocytose that particle. So the solid waste is removed with the help of lysosomes. Whereas peroxisomes, if you have a liquid droplet like a toxin or something which the cell doesn't need and is harmful, the peroxisome will come, take it inside and get it destroyed. So peroxisomes are helpful in removing liquid waste or toxins. Some could be waste, some could be harmful to the cell if they are left alone. Mitochondria, the next organelle that looks like this, with some membranous folds on the inside. Mitochondrion, singular, mitochondria, plural. They are considered to be the powerhouses of the cell, meaning they produce all the energy needed by the cell in the form of what? ATP. Hopefully by now you know what is ATP. Not only what does it stand for, what is ATP, ADP, AMP, how we make ATP, where we make ATP, where most of ATP is made, which is the mitochondria. Some ATP is formed in the cytosol when the glucose breakdown occurs, glycolysis, but then when you do the aerobic respiration, and the pyruvic acid is passed through the citric acid cycle, you produce eventually some compounds that are taken to the mitochondria and processed to give most of your ATP. So you produce 2 ATP per glucose in the cytosol, and you produce per glucose 36 ATP in the mitochondria. Now you may have some little tiny droplets of granules or pigments. They are called inclusions. Some cells may have them. That kind of wraps up all the organelles and their function. So there's a summary in table 3-4 if you want to take a look. So when you go through the table, if you have understood the chapter, then that will be helpful to recall the information. I'll give you a couple of minutes. In a few minutes we will start on chapter 4. The chapter 4 deals with the DNA, transcription, translation, how proteins are formed, and some genetic defects. If you have some damage or improper alignment of chromosomes, and how cells divide.